Hi, my name's Christopher James Ward from the Kansas University Medical Center, and today I want to talk about the use of Cas12a RNA complexes to knock out genes in renal cortical tubular epithelial cells, RCTE cells. Now, RCTE cells are male human renal epithelial cells, and they're an immortalized line originally isolated in the Cas12a lab. We use them because, first of all, they're human, as we said, they're ciliated, and they make considerable amounts of polycystin-1, the product of the polycystic kidney disease type 1 gene, so that's autosomal dominant, and significant amounts of polycystin-2, the product of the autosomal dominant polycystic kidney disease type 2 gene. And they have detectable amounts of fibrocystin, which is a product of the autosomal recessive polycystic kidney disease gene. And the name of this gene is polycystic kidney and hepatic disease 1. So that's PKHD1. So one of the problems we've got in PKD is that we have antibodies to these three proteins, and we're sure that they work on Western blot. But when we use them in immunofluorescence, we don't have adequate human negative controls. So the plan here is to either delete or frame these three proteins in the context of this cell line. So what are we actually going to do? We're going to use the RCTE cell line because it's immortalized and it grows very well, and it's easy to electroporate, and of course it makes our proteins. And this talk is going to be in two parts. The first part will be dealing with the technicalities, and the second part will be dealing with the analysis of the clones that we get. So the overall strategy is that we'll use Cas12a protein from Acetaminophococcus species BV3L6. Now, this has been modified to enhance its activity at 37 degrees, and it has optimized nuclear localization sequences. The protospacer adjacent motif, the PAM sequence, is TTV, where V is not U and it's not T, so it can be either A or C or G, okay? And so that's a critical thing. All Cas12a's sites must begin with that 5' to 3'. The Cas12a molecule will cut DNA 18 to 19 base pairs from the 3' end of the PAM and 23 base pairs from the PAM on the opposite strand, leading to a staggered cut with a 5 base pair 5' overhang. Now the strategy we're going to use is we're going to try to utilize microhomology end joining. And so the idea is to have two short segments of direct repeats on either side of the cut site. These have to be greater than 3 base pairs in size, and will be substrates for the microhomology end joining after Cas12a cuts between the two direct repeats. Now what we're hoping is that the two direct repeats then recombine by microhomology end joining and delete the intervening sequence. So if the length of that sequence is not divisible by 3, supposing it's 10 or it's 13, then that will frame the gene if the Cas12a site is within an exon. So how do we do this? Well, actually, it's quite an intense computational task, and we use the GeneSculpt org Menfu program. And so it will search your favorite gene, as long as you have the GenBank or EMBL accessions, and it will look in the exons and find Cas12a sites with microhomologies on either side of the cut sites. You can also look at other CRISPR systems, for example Cas9. And so we use this website to find our Cas9 target sites and our microhomologies. Okay, so that's quite complicated. it's better if we actually look at a diagram and things become much simpler. So what we're going to do is we're going to be targeting the PKD2 gene using Cas12 and a guide RNA, a gRNA. Now, the MEMFU program selected a target in exon 7 in PKD2, and so that's got two nice microhomologies, as we'll see in a minute, and it has a Cas12a site that will cut between them. So first thing we've got to do is we've got to synthesize a guide RNA. So there's a constant part of every guide RNA from 5' to 3' that is involved, it's called the scaffold, and is involved in binding the RNA in an appropriate way to the Cas12a protein. Okay, and so that's in green here. Now notice that these are ribonucleotides, and it's RNA because instead of T's we've got uracils here. Then in blue we've got the actual sequence that will target. Okay, so the plan is we make a complex between Cas12a and the guide RNA in a test tube. Then we electroporate that into RCT cells. The nuclear localization sites on the Cas12a take the complex into the nucleus, and then this sequence targets it to PKD2. So let's look at the target site. We've got a PAM here, T, 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 C. So that fits the consensus T, 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 V. And then if you look here, the uracil maps to the thymidine, the G to the G, the G to the G, the A to the A, the A to the A, the G to the G, the A to the A, and the U to the T. And it goes on and on and on, and the homology stops just here after the C. Okay. Now the Cas12a will cut at position just after position 18 and just after 23 on the other side. And so you'll have this as an overhang, which will be A, G, 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 G. Okay. That's not that important. What is important are the microhomologies. These are direct repeats. A, C, T, T, A, C, T, T. Now, what we're hoping is that the Cas12a will come in here and cut. And it might go through multiple rounds of cutting where the DNA is just repaired and repaired perfectly. The cells are very good at repairing their DNA. But there is a pathway that uses microhomology end joining, which will recombine this with this and delete the intervening sequence. And that's 10 base pairs long. And so you'll end up with this. So this has got two effects. One is it will destroy the site for the guide RNA, so it will stop cutting here. And the second thing is it'll delete 10 base pairs. So 10 is not divisible by 3. 3 goes into 10 3 times, give you 9, remainder 1. And so this will then frame the protein. And so that's the plan. Now, if we frame PKD2 here, what will happen is that the binding sites for the two antibodies that we're going to be looking at, which are D3 and YCE2, will cease to exist. You know, if it's a homozygous for this change. And so we will have a wild type cell where you'll have nice ER staining, and you'll have a mutant cell that doesn't have PKD2, and the antibodies won't bind to it. And so that will verify these antibodies. Now we're going to do this for PKD1 and PKHD1. We've already done that. I'm also going to do it for CU62 because I've got 2 monoclonals for that one, and I really need to make sure that they're cognate to their target. I'm pretty sure they are, but you've got to make sure. So here's an overview of the protocol. We plan to use Cas12a CRISPR complex instead of a DNA construct to disrupt the target gene in renal cortical tubular cells. The complex is introduced by standard electroporation using a neon system from Thermo Fisher Scientific. This is an advantage that no exogenous DNA is placed in the cell, and the time for CRISPR exposure should be short. The CRISPR RNA itself is short in length here, it's about 40 base pairs, and it can be synthesized chemically. And so we get ours from IDTE, and the CRISPRs cost $105 for 2 nanomolar, and 2 nanomolar is enough to do many, many experiments, as we'll see. So these are the materials that are required. We need the Cas12a CPF1 Ultra protein. We need an electroporation enhancer. We don't know really what's in there, but it really helps. We need 2 nanomolar of CRISPR-Cas12 Cas12a CRISPR RNA from the URL above. I have a neon system, which I inherited, and I buy a neon transfection kit, a 10 microliter kit. Now, here's an important thing. I actually buy in my TE solution for RNA work. The IDTE solution is expensive, but it's RNase free, and I've had no trouble with it. So the protocol is we grow the RCTE cells to 80% confidence in RPMI 1640, pen and strep, and 5% fetal calf serum. Now, if you can't afford fetal calf serum, it's alright to use bovine serum at the same concentration. Newborn bovine serum. It's very cheap. While harvesting, make up the 2 nanomolar CRISPR in 27 microliters of IDTE. This gives you a 75 micromolar solution. Combine 5 microliters of the CRISPR, 5 microliters of the Cas12a protein, ultra, in an RNase-free 1.5 ml tube and incubate at room temperature for 20 minutes. Now, by RNase-free, and this should be a ml, RNase-free, I mean taken straight from the package and autoclaved. After incubation, add 10 microliters of the ALT-R CPF1 electroporation enhancer at 10.8 micromolar. And this is designated the final mix. Okay, so you've got your Cas12, you've got your CRISPR, and you've got some carrier, electroporation carrier in this tube. And you're just going to add your cells to this. So we have to harvest the cells using trypsin EDTA and neutralize the media with fetal calf serum. Then we spin about 500 Gs for 5 minutes and wash 2 times in PBS. You'd want to be quite gentle with the cells. And then count them and then take them up in neon buffer R at 5 times 10 to the 6 per mil. And the cells are quite cloudy. It looks like milk actually. There's a lot of cells in there. So in preparation for the electroporation, you warm up your RPMI 1640 pen and strep fetal calf serum and 6 well plates, and so it's nice and warm. Add 40 microliters of cells at 5 times 10 to the 6 per mil in buffer R to the final mix, and then electroporate using the neon system, and we use 1650 volts for 20 milliseconds and 1 pulse. Then you add the 10 microliters of cells that's in your pipette to a well in a 6 well plate and incubate, and you do this 5 times. And as a control, we electroporate 10 microliters of cells with 1 microgram of GFP plasmid, and we use a PMax of GFP to get an idea of efficiency. Efficiency is usually really good. It's at least 80%. And then we'll let these cells just recover for 3 days in a CO2 incubator at 37 degrees. And then harvest the cells using trypsin EDTA. And then dilute the whole lot, all 5 wells, into 50 mils. And then plate out 100 microliters per well into 496 well plates. If there's a lot of cells down there, you can do a 1 in 10 dilution. But usually this is limiting dilution. There's not a lot of cells that kind of survive this and sit down. And so now we'll go into the lab and we'll do some practical work. And thank you for listening to me. Hi. So today we're going to make up some Cas12a CRISPRs and then complex them with Cas12, okay, 12a, and then just leave them for 20 minutes to make a complex. And then while we're doing that, we'll get our cells ready and put them at the right concentration, which is 5 times 10 to the 6 into buffer R, and then we'll electroporate the Cas12 CRISPR Cas12 complex into the cells and then plate them out. So first we've got to make up the Cas12a CRISPRs, and so I've got 2, one for a protein called CU62, another one for a protein called C16, and we need to bring them up to 75 micromolar. And so to do that, we've got to take up the 2 nanomoles that we've got here in 26 26.6 microliters of TE buffer. In fact, this is IDTE buffer. 26.6. Right. So the good thing is that the RNA molecules, the CRISPRs themselves, go into solution very, very quickly. And nice and short. So here we go. Just put the lid back on. So that's C16. This is CU62. Okay. And here we have our Cas12a, and it's CPF1 Ultra. So it's a specially genetically modified Cas12a, which has got 2 nuclear localization sites on it. And so the idea is to actually make a complex between the RNA and the nuclease in these tubes here, and then apply them to the cells. So we'll do this in the hopefully that's gone into solution. Flip around. So the good thing is it's relatively soluble. So I'm going to use 5 microliters of the Cas enzyme for each. I'm going to get this back onto ice as quickly as possible. It's 5 microliters. And at the bottom. That's the enzyme. It doesn't have the RNA attached to it yet. C16. 5 microliters of this. Straight to the bottom, head it up and down. And then again to the CU. And leave that for at least 20 minutes. So C16, C16, C, good. And here is the buffer. It's IDTE buffer, page 7.5. Just leave that, and then we'll take these back and freeze them. Okay, thank you. So the next step, we've got our Cas12a complex and RNA together for both the genes that we're going to knock out. What we're going to do is we're going to knock out the genes in these RCTE cells. So these are renal cortical
tubule epithelial cells, originally from Professor Jing Zhu's lab. They're nice cells in that they're human and they have primary cilia. They don't form tight junctions, but they do have a lot of polycystin, and Cu62 and C16 are potential ligands. So what we want to do is basically knock these genes out using the Cas12 and the neotransfixion system, and basically knock them out and use them as targets for our various monoclonal antibodies that we made. So if we knock out the genes in these, there will be negative controls for the monoclonals. So I'll get on and I'll wash these and basically elute them with, get them off the plastic with trypsin EDTA, and then get them in suspension, spin them down and get them up to PBS, and then we'll count them. So I'll do that right now. There's a neon system here, and I'll just switch it on at the back. And here you can see we've got to program it, and what we're going to do is give it 1650 volts for 20 milliseconds, and we're only going to do it once, okay? And then we're going to set up the electroporator and get a ring ready for the main event, which will be the electroporation. I'll show you how to do that. So we just wait for it to warm up. This is a neon system, and so what we want to do is we want to reprogram this. So it's 1650 volts, done. 20 milliseconds for one pulse, okay? And that's what we use for the RCTE cells, and we use buffer R. So around this side, we're going to open a new package here and put in a new electroporation cell, and these are sterile, so I always use a knife to get in there. And at the bottom of the package like this, and just ease one out. Bottom first, grab it, get it out, okay? And then it goes in like this into the neon system, okay? And I'll leave that like that, and then you've got to seal your bag. I just use autoclave tape. Maybe I should have had the autoclave tape ready. Let's see there. To get it started here. And we'll just seal the bag. Seal it. Handle it over. Seal it. Put it away somewhere safe. Okay. Now we need to put 3 mils of buffer E in here. Put a neon electroporation buffer E. One, two, three. Put that right down to the bottom, like that, and split it in. Just make sure it's in. And it's in. Good. So that's the machine ready. I think we're going to put a bit more tape on there. What we've done is we've trypsinized the adherent RCTE cells off the plastic and recovered them. They've got a nice pellet there. There's about 7 to 10 million cells there. So we need to quantify them. So the first thing we're going to do is just tip off the supernatant and take this up in 10 mils of PBS. And then we're going to count them, do a trypan blue. And then we're going to spin them down again and take them up in PBS again, and then take some of it and put it into epinephrine. Very quickly spin that down and take it up in buffer R for electroporation. And we need a concentration of about 5 times 10 to the 6 cells per mil in the buffer R. Okay, so there's a nice pellet there. Okay, so I'm just going to tip, be very gentle here, and just tip. And then you got your pellet, and then you just break it up like that, very gentle. I want to be as gentle as possible here. And then I'm going to take this up in 10 mils, so it should be about 1,000,000 a mil, hopefully. We'll see, because we're going to count them. Okay, quite gentle. Right, so we have a machine that can count cells, and it uses a plastic hemocytometer like this. And what we need is we need a tube. This bit doesn't really have to be sterile. I'll re-autoclave these in a second. Right, so this is filtered trypan blue, and I always filter it because for some reason I get bits in it, and that can interfere with the counting. And so I want 100 microliters of cells to count. You can get nice accurate 100 microliters. So again, give these guys a little mix. And then I want to go in and get 100 microliters as well. Without touching the sides if we can. Like that. So that's good. That looks like about 1,000,000 a mil to me. Just I have experience. We'll see in a minute. Because we would need to get this rather accurate. Counting them by hand, I found it as being completely inaccurate. As Ben Veniste found out to his cost many years ago. You just pipette these up and down. Now, when you look at this hemocytometer, we just need to slot it in like this, watch, and then like that. And then the other side, like that. Okay, there we go. Now we're going to take this upstairs and we're going to get an accurate count. And we'll need a lab boot to write it down. That lab boot. And we should be alright. Okay, so here we're going to use a cell counter. It's a Biorad PC20. Switch it on. Okay. And then these cassettes just slot straight into it. It just says count cells, it's automatic. It's focusing. Oh goodness, pen. Then we've got to write this down. So we need to view each from the laptop. So what's today's date again? Today is 18th. Right now, so 9, 18, 22. First one. So it's detected the trypan blue. And that tells you how many cells are alive and how many are dead. If it takes up the trypan blue, they're dead. Okay? If it doesn't, then they're alive. So it'll give us a live count, live dead count, as well as the total number of cells. Right, so we've got 1.22 times 10 to the 6 on this one. And it's 100% viable, eh? That's good. I think it's because I filtered the trypan blue, actually. You always have to do that. Put it for a 0.2 microns. So we do it twice, just to see, because there's always going to be sampling error. And then we'll take the difference. We'll take the average. Okay. So Tri-Band Blue is carcinogenic dye. You've got to be quite careful with it. So it's 1.27 this time, and 97%. We'll call it 100%. All right. So we take the average of the two, 1.27 plus 1.22 equals divided by 2. So that means we've got about 1.25 times 10 to the 6 per mil in the PBS. Now if we want to concentrate this up, okay, to 5 million a mil, okay, we'll have to concentrate it 5 divided by 4 times. So that's nice and easy. So what we can do is we'll pellet them again, wash them back into PBS, and then we'll take the appropriate amount to give us probably 200 microliters at 5 times 10 to the 6. Okay, so we'll do the sums for that. So that's very simple. If you want 200 microliters at 5 times 10 to the 6, you need 800 microliters of these cells. Okay, so that's pretty easy. So we can do that. But first I want to give them another wash in the PBS, because that helps to get rid of any RNases. And then we'll gently spin them down in an Eppendorf and take them up in the buffer R for the electroporation. Good. Now we've got our 2 complexes here. We've got to add electroporation enhancer. We don't really know what's in there, but we're going to use 10 microliters each. Okay, and this will enhance the electroporation. So what we need is these guys here. Okay. Right, there we go. There we go. C16. See you. Down to the bottom and squeeze it in. Okay, that's one done. There's the other. Now we want about 200 microliters of cells at 5 times 10 to the 6, okay, in buffer R, okay. So what we'll do is we'll take these cells and we'll bring them up in 5 mils, and that will bring them to 2.5 times 10 to the 6, and then we'll take about 400 microliters of them and spin them down, and then take them up to 200 microliters of buffer R. And then each of these will then take 40 microliters, mix them quickly, and then we'll electroporate them, okay? So that's the plan. Right, so what we're going to do here is we're going to rescue these cells again. Again, I'm just going to tip off the PBS, and then I'm going to take it up in 5 mils, which will double the cell concentration. And the washing with PBS seems to reduce the amount of nucleases around, makes a better electroporation, okay? More efficient. So we'll take up in 5. And then if I want 100 microliters of cells at 500,000, 5 million a mil, okay, I just have to take 200 of this and quickly spin it down, and we'll do that in the main lab. final wash into PBS. It's really nice. Okay. So I want final 100 microliters at 5 times 10 to the 6. So I'm going to need 200 microliters of these cells. I'm going to put them in here and I'll quickly spin them. Very gently, but quickly spin them. And then take them up in 100 of buffer R. And they're ready for the electroporation. So let's get 200 of these. Right. There you go. What we want 100 microliters of cells in buffer R, okay? And so we've got 200 microliters of cells here at 2.5 million a mil. We spin them down and take them up in 100 of buffer R. We'll be all right. Just a quick one up and down. Now you can see your cells there. Okay, so we're going to go and take those up in the buffer off. Let's go. All I got to do now is take that buffer off very carefully, not disturbing the pellet too much. There we go. And just gently break up that pellet again. And we're going to just take 100 of R. This is buffer R, the electroporation buffer from the neon kit. Fiddly, eh? Good, there's 100. And just mix. Okay, good. There's our mixture. Now, you got to kind of speed things up a little bit now, because they don't like it being in the intracellular buffer here. So we'll go with the C16 first. And we're going to add 40 microliters. Like that. Mix it a couple of times. Right. That's the first one. Okay. Now, in the incubator, I've got some more media in place. I'm going to use this. Alright. This is going to be C16. We'll do this one first, okay? Now, what we need to do is we've got these neon tips come with the neon kit. So we open them up. We've got 4 tips. You need a special pipette here. Put it down like this. Grab. Right? You see that? Now, what we're going to do is we're going to take up 10 microliters without getting a bubble. Be tricky. Like that. Because that's a mixture of your CRISPR Cas12a and your cells. And you pop it in like this, right down, and then press the button. Okay, tell me if you can press the button. Good. Have you done it?
Yep.
Okay, good. So that's the first one done, and you just pop it in there. All right. Next up, just go as quick as you can and try to reuse everything. Okay. If there's a misfire, you can always mark the well that's misfired and not use that. Good? Thank you, Tommy. So Tommy's just pressing the button for me. Again. Shoot. See that?
Yep.
Good. We're doing good here. Now, at the very end here, what we're going to do is we're going to do a DNA control, just to make sure that the whole thing's work. Right, next part. So far so good, we haven't had any misfires. Must be doing something right. Imagine what it is, but then we'll try. And again, fire. We'll see if there's enough for one last one from here. Sometimes there is. Oh, there is. Alright, man. Oh no, it's got a bubble. Oh, it's got a bubble. Ah, too bad. Oh well. And you get reuse it like that. Okay. Next up is CU 62. I'm trying to write a paper on. It's taking me a while to really understand this molecule. And I think I'm there. Alright. Okay. 40. Mix it and now put this away. Oh shoot, I just threw that one away. Sorry about that. Bit of a waste. Okay, good. Right, now see you. A little bit worried about that one. I think I touched the top by accident. I should have done that. Just mark that just in case it gets bugged or something. That was a bit of a mess up. I just touched the top of it. I'm not sure if that's completely sterile. Okay, fine.
Tip connection error.
Okay. Sorry. Let's just try again. Might be. Oh yeah. See, there's a bubble. Yeah, I've got it now. Yeah, there was airspace in there. Okay, fire again. Sorry. Good?
Yep.
Yeah, it was just an airspace, yeah. Wasn't completely the circuit. Okay, fire again. Good.
Yep.
And, no. Once more. Go. Okay, fire. Did it say anything?
It said it was completed. It was good.
Sometimes, let's see if we can get enough for the last one here. Oh yeah. We get a bonus one this time. Okay, fire again. Cool. Again, now there's one last one we need to do. It's control. Okay, and it's a GFP control. So what I do is, we've got about 20 microliters in there. I have got a GFP control. It's from an Amaxa kit, and it's just an Amaxa plasmid at 0.5 micrograms a mil. So I'm actually going to use about 2 microliters of this into 20 microliters of residual buffer R that we've got here, and then I'm just going to electroporate it very quickly. I just want to make sure that, you know, the protocol worked, and I'm not going crazy here. It's just a only straightforward plasmid, GFP plasmid. Something like that. The DNA's in there, give it a quick click. Now then, what we're going to do, I'm going to put in this one here. I'll just leave that in there for a minute. And leave it as it is. Now then, I've got one last guy. Put them up. I bet the DNA one doesn't work now. This is DNA control. This should make the cells go green because there's a GFP, green fluorescent protein, Victoria aquaphora. There we go. And we pop that in like this. Okay, Tommy. Cool. Right, we've got control now. Right, okay. Now, the best thing to do is just have a quick look at the cells, make sure that their cells are, you know. Yeah, they look really good. I'll look at the GFP, because sometimes the DNA is a bit rougher than the actual, oh yeah, DNA is a bit rougher than this guy. Yeah. Well, if it was going to be a cell, I'd prefer to have the Cas12a complex rather than the DNA. The DNA seems to kind of hurt them. Okay, good. Finished. Hi, so here are some GFP positive RCTE cells. This is not a very good electroporation. Usually
they're 80 to 90% positive. Here they look about 40 to 50. It's still good enough to go on and work with your Cas12a CRISPR electroporation. Thank you.